In 1773, in a small room somewhere in Germany, a pastor pressed a clump of dried moss against a glass slide and wrung from it a single drop of water. The drop was cloudy. It held almost nothing visible to the naked eye, just the faint color of whatever the moss had given up when squeezed. Johann August Ephraim Goes placed this drop beneath the lens of his microscope, adjusted the brass tube, and looked. What he saw moved, not quickly, not in any way that suggested urgency or alarm. The movement was deliberate and rolling, a slow progress across the glass that reminded him, in some quality he could not immediately name, of a bear navigating uneven ground. 8 legs, a rounded, plump body, a gait that placed each leg with what appeared, at this magnification, to be some small degree of consideration. The animal was shorter than the width of a human hair. It seemed entirely unconcerned with the unusual circumstance of being observed. Goise had no name for it yet. He was a man who spent much of his professional life attending to parish matters, and the rest of it attending to natural history, a combination that was not unusual for the educated men of his century. He collected and described creatures that other naturalists had overlooked, organisms too small or too humble to attract systematic attention. The drop of moss water had been routine. The animal walking through it was not. He called it Kleiner Wasserbeer, the little water bear. It was the right name for the wrong reasons, or perhaps the right name for reasons he could not fully articulate, because what he saw was not a bear in any structural sense, but something in the quality of its movement, that particular, unhurried wait, connected in his mind to a large animal crossing difficult ground. The name was informal, descriptive, personal. It stuck anyway, and it has been used in one language or another for the more than 250 years since. What Gose had found, pressing moisture from dried moss in the ordinary course of his naturalist's work, was something that had been living in the thin films of water around terrestrial mosses and lichens for as long as mosses and lichens had been there, which is to say, for a very long time. He had not discovered it in any meaningful sense. He had noticed it, which is a different thing. The animal had been present in that moss, and in every clump of moss like it, for longer than any record that could be checked. The lens Gose used that day was a modest instrument by any standard, a tube of brass with ground glass at each end, the kind of microscope that was becoming more accessible in the mid-18th century, as lens making improved, but was still far from what a modern researcher would recognize as adequate. What it could do was enough. The animal he was looking at was, at its largest, about 1.5 millimeters in length. Most were considerably smaller. The one crossing his glass slide may have been somewhere in the middle of that range, visible if he knew where to look, and the light was right, but invisible to anyone not specifically searching. It measured between 1/10 of a millimeter and 1.5 millimeters, depending on species and life stage. This is a range that begins at roughly the size of a dust particle and ends at something that could, with careful attention, be spotted with the naked eye on a bright surface, though it would look like almost nothing, a speck, a mote, easily ignored. Through Gose's lens, at the magnification he could achieve, it was recognizably an animal, 8 legs, a defined head, a body that was plump and segmented in a mild way, and that gait, rolling and slow and peculiarly deliberate. The legs themselves were stubby, more like protrusions than true limbs in the way an insect's legs are limbs. They extended from the underside of the body in pairs, 4 pairs in total, with the hindmost pair pointing slightly backward. The tips of those legs varied considerably, depending on which species happened to be under a given researcher's lens. In some, the feet ended in clusters of small claws, curved and precise. In others, the feet bore adhesive pads or discs that allowed the animal to cling to surfaces without gripping in any active sense, simply resting against them with enough contact to hold. Both strategies worked well in the wet, fine-textured world these animals inhabited, where the surfaces were the walls of plant cells and the fibers of moss leaves and the rough, porous faces of lichen. Goas's initial description was not a formal scientific account in the way that term would later be understood. It was an observation, carefully noted by a man who found the creature genuinely worth recording. The formal scientific work of naming and classifying the animal came partly from him and was extended and refined by others over the decades that followed. Among them was an Italian biologist named Lazzaro Spallanzani, who encountered the same creature and was moved by a similar quality in its movement. Spallanzani described its pace as that of a slow stepper. From this impression, from the Latin root for slow, he derived the name that became the scientific designation for the group, Il Tardigrado, the slow stepper, Tardigrada. Both names have survived. In scientific literature, Tardigrada is the phylum designation, formal and precise. In popular writing and in conversation, and in the notes of anyone who has first encountered one of these animals through a microscope lens and found themselves looking at something that moved with the unhurried confidence of an animal entirely at home in its world, the little water bear persists just as well. The world these animals called home was specific and once described, immediately recognizable as something most people had walked past without knowing it was there. Tardigrades lived most commonly in thin films of water, not pools, not rivers, not even puddles in any conventional sense, but the microscopic layer of moisture that formed around individual moss leaves and lichen thalli when the air was damp enough to allow it. This film was sometimes only a few cells thick. It existed in the space between one moss frond and the next, in the gaps between lichen crusts on stone, in the tiny channels formed by the structures of the plant itself. From inside that film, a tardigrade had everything it needed: water, organic material, other microscopic organisms on which some species fed. The diet of a tardigrade depended on species, but the general range included plant cells, which some species punctured and emptied using a set of internal stylets, sharp, needle-like structures used to breach cell walls, and algae and single-celled organisms, and in the case of some predatory species, other small invertebrates, including other tardigrades. This was not a simple animal in its feeding behavior, even if its body plan was compact. It had internal organs. It had a complete digestive tract. It had a nervous system, simple but present. What it did not have was a circulatory system with a heart in the conventional sense, or lungs, or any of the apparatus that makes the bodies of larger animals so expensive to maintain. A moss-covered garden wall, looked at from the outside, is just a wall with something green on it. And looked at through a hand lens, it becomes a landscape of tiny upright fronds, some dry and contracted at the tips, others still damp and extended, the whole surface shifting between states as the light and air changed through the day. Looked at through a microscope, that same surface revealed a world in motion: algae, bacteria, the small transparent bodies of rotifers, and among them, moving at its characteristic rolling pace, the round-bodied, eight-legged animal that had been there all along. Tardigrades lived in the moss on garden walls. They lived in the lichen on headstones and forest boulders. They lived in the damp substrate of roof tiles and the sheltered microhabitats of drain pipes and windowsills. Anywhere water collected in small enough amounts and plant life of some kind took hold, tardigrades were a reasonable expectation. This made their distribution remarkably broad. Not because tardigrades were adapted to a range of dramatically different habitats, but because the specific microhabitat they required, a thin film of water around a moss or lichen, was itself found almost everywhere. The conditions that produced it were common: moisture, plant life, a surface to grow on, a rock face in a temperate forest, and the stone of a churchyard wall, and the tiles of a greenhouse roof, all offered the same essential resource, scaled down to the level at which tardigrades operated. Across this distribution, researchers over the more than 2 centuries since Goy's first note had collected and examined specimens from every continent on Earth. Not every country, not every habitat type, but every continent, and the inclusion of Antarctica in that list was not a formality. Antarctic specimens were not collected from warm, sheltered habitats by researchers comfortable in the field. They came from glacial mosses and soil samples taken from the transantarctic mountains, from coastal moss beds exposed to the brief and cold polar daylight, from sites where the conditions for most life were, by any common understanding, severe. Viable tardigrade specimens were recovered from all of them. The animals found there were not different in any fundamental way from those found on a piece of garden lichen in a temperate European city. They had the same basic body plan. They lived in the same thin film of water around the same low-growing plant life. They were simply doing so at a latitude and in a climate that most organisms decline to inhabit. The question of how they had come to be so widely distributed was one that the scientific study of tardigrades had addressed in several ways. The most plausible account involved passive dispersal. Tardigrades, particularly in their desiccated state, were light enough to be carried by wind, water, or animals across considerable distances. A particle of dried moss lifted from a rooftop by a gust of wind might carry tardigrades with it. Migrating birds moving between continents could transport tardigrade-bearing material on their feet or feathers without any deliberate mechanism of dispersal on anyone's part. The animals required no active role in this process. They simply needed to be present in a substrate that could be moved, and they were. This passive quality extended to their general relationship with the world they inhabited. Tardigrades did not actively seek new territories. They did not migrate. They did not appear to compete for space in any aggressive sense. They occupied the film of water around their particular piece of moss, fed on what was available, reproduced when conditions allowed, and otherwise continued their slow, rolling existence in a manner that suggested from the outside a creature of considerable patience and limited ambition. This was not quite right as a description of their biology. Patience implies waiting for something, and the tardigrade's relationship with waiting was considerably more complex, but as an impression of how they appeared and behaved in ordinary conditions, it was not far wrong. The group to which tardigrades belonged, the phylum Tardigrada, contained, by the time researchers had worked through the accumulated species descriptions of more than 2 centuries, over 1,300 distinct species. These were formal descriptions, each one supported by morphological examination, and increasingly by genetic analysis, each one representing a population of animals that differed from all others in the specific combination of physical features, and were examined. Genetic markers that distinguished one tardigrade species from another. 1,300 is a considerable number for a phylum whose members all shared the same basic design: 8 legs, a rounded body, a set of oral structures for feeding, and a life lived in a film of water. The phylum was divided across three classes. Eutardigrada contained species that lacked certain ancestral sensory structures found in other classes, and was itself divided into orders and families reflecting the diversity within the group. Heterotardigrada retained more ancestral features, including elaborate head structures in some marine species, and represented a distinct evolutionary lineage that had diverged early in the group's history. These two classes were well established, and their boundaries supported by both morphology and molecular evidence, and new species within both continued to be described on a consistent basis, not a flood of new discoveries, but a steady accumulation, a few formal descriptions per year, each one requiring a researcher to have noticed something small and wet that slightly differed from everything already cataloged. The third class, Mesotardigrada, occupied a more uncertain position in the literature. It had been described from a single specimen collected from a hot spring in Japan, an animal that differed enough from the existing classes to be assigned its own. The specimen itself had since been lost, not a common outcome in natural history collections, but not entirely unheard of either, particularly for a specimen so small that a moment's inattention could make it effectively irretrievable. Without the specimen, and without subsequent collections from the same site, the class remained formally described, but practically inaccessible to further study. Its existence in the literature was a placeholder of sorts, a label for something that may or may not have been what the original description suggested it was, waiting in taxonomic suspension until someone found another specimen or the record was revised. The business of describing new tardigrade species was, by the standards of natural history, a patient occupation. It required access to a microscope of sufficient power, the ability to collect and prepare specimens without losing or damaging them at a scale that made handling genuinely difficult, familiarity with the published descriptions of existing species against which any new candidate had to be compared, and the particular willingness to spend extended periods examining small animals. And researchers who did this work over the course of a career produced formal species descriptions that accumulated in specialist journals, each one adding a name and a set of distinguishing features to the total catalog of known tardigrade diversity. What that catalog represented, taken as a whole, was not so much a collection of dramatically different organisms as a register of variation on a single theme. The basic tardigrade body plan was conservative. 8 legs, a rounded head, a body that was slightly longer than it was wide, a set of internal structures for feeding and digestion, a cuticle that was shed periodically as the animal grew. This plan appeared, with modifications, across all 1,300 described species, across all known environments in which tardigrades had been found, across the full range of the animal's documented distribution. The variations between species were real and meaningful to researchers. Different claw shapes, different head structures, different numbers and arrangements of sensory organs, but to a non-specialist looking through a microscope for the first time, one tardigrade was recognizably kin to another. This recognizability across species was itself a feature of tardigrade biology worth noting. Many animal groups, as they diversified over evolutionary time, produced forms that varied so substantially from their common ancestor that the relationship required genetic analysis to confirm. The vertebrates, for instance, included both a lamprey and a blue whale within the same phylum, a range of forms that bore little obvious physical resemblance to each other. The tardigrades had, by contrast, maintained something close to their fundamental body plan across the full range of their diversity. The animal that Goisy observed through his brass-tubed lens in 1773 would have been recognizable in its basic features to a researcher examining a different species from a different continent more than 2 centuries later. Part of what made this recognizability possible was the specific set of conditions to which tardigrades were adapted. The thin film of water around a moss frond was a particular environment with particular demands, and an animal well suited to it did not necessarily benefit from major structural changes. What worked in damp moss in northern Germany worked in damp moss in coastal Antarctica, which worked in damp lichen on a rooftop in Japan. The habitat was common enough and stable enough in its basic character that the body plan adapted to it could remain largely unchanged across a very long distributional range. This did not mean tardigrades were simple. The 1,300 described species represented real biological diversity, real differences in feeding strategy and habitat preference and reproductive behavior. Some species lived primarily in freshwater sediments rather than terrestrial mosses. Some were marine, living in the spaces between sand grains on ocean floors, and their body plans reflected adaptations to that specific environment. More elaborate head structures in some cases, different claw arrangements, bodies shaped by the particular demands of navigating a habitat that was consistently wet rather than prone to drying. The diversity within the phylum was genuine. The conservatism of the basic form was also genuine. Both things were true at once. A natural history collection somewhere, and there were several that held tardigrade slides as part of their reference material, might have contained, in a single drawer, mounted specimens representing dozens of species from a dozen countries. The slides would have been small, labeled in careful handwriting, or in more recent ones, printed labels placed beneath the cover slip. Each slide held, in a drop of mounting medium, an animal the length of a large grain of fine sand, or smaller, its body preserved in some particular posture, its legs extended or retracted, its internal structures visible to varying degrees depending on the preparation method and the optical quality of the equipment used to examine it. Looked at as a collection, those slides represented something beyond the individual specimens. They were a document of distribution, this species from a moss sample in Norway, that one from a sediment core from the Pacific floor, another from a lichen collected by a field team in the mountains of South Africa. Together they described a creature that had, over a very long time, established itself in the thin films of water at the edges of nearly every terrestrial and freshwater habitat on the planet. Not conspicuously, not with any fanfare visible from outside, simply by being suited to a widespread and common resource and reproducing successfully in it wherever the resource was found. The green moss of a garden wall, the gray-green lichen of a forest stone, the sparse coastal mosses of a high-latitude field site in Antarctica, all of them held, within the film of water, their surface structures retained, a population of animals moving at the same slow, rolling pace, feeding, reproducing, going about the ordinary business of existence at a scale most observers never considered. Goizzi had considered it one afternoon in 1773, pressing moisture from a dried clump of moss and placing the result under his lens. The animal he saw moving across the glass had been there already, before the lens was placed, before the water was collected, before the moss was removed from wherever it had been growing. It would have been there still had he simply walked past. The tardigrade, doing its slow work at the film of water's edge, asked for no particular attention. It required only the moss and the moisture and the ordinary passage of time. Along the coasts of Antarctica, in the sparse mosses growing in the brief warmth of the polar summer, the same animal conducted the same unhurried life. The cold around it was genuine, the exposure considerable, the habitat as stripped back as any terrestrial landscape on the planet. In the water that remained, thin and small and sufficient. The eight-legged animal moved, fed, rested, and moved again, small and unhurried, in a place where almost nothing else moved at all. On the surface of a clump of moss, the water is going away. It goes slowly at first. The thin film that coats each moss cell growing shallower, the spaces between the fine green filaments drawing back, the particular liquid world that a tardigrade moves through, becoming less than it was. The tardigrade registers this, not with anything that resembles thought, but in the way that a cell registers a change in its immediate chemistry, the way a system built around the presence of water notices with the whole of itself when that water begins to leave, and so the legs begin to draw inward. They go one pair at a time, or several together, folding toward the body with a slowness that matches the slowness of the drawing. The tardigrade's body, which a moment before was extended, moving, occupied with its small work of feeding and walking, begins to contract. The rounded head pulls back. The sides compress. What was a blunt, eight-legged animal, roughly the shape of a very small bear, becomes something rounder, more compact, a form that is tending toward a closed thing, a contained thing, a thing that has gathered itself in from the edges. By the time the water film has grown too thin to move through, the legs have disappeared entirely, tucked inside a surface that is now smooth and sealed and very still. What the tardigrade becomes in this process is called a tun. The word is old. It once meant a large barrel or cask, the kind used to store wine or ale, the kind with curved staves and a tight circular form. The name fits. The dried tardigrade in this state is a tiny barrel, closed at every surface, sitting on the drying moss with no movement and no visible sign that anything inside it is alive. It can stay this way for years. Under the right conditions, cool, dry, undisturbed, it can stay this way for decades. The moss it sits on might be carried indoors on a shoe, stored in a desk drawer, pressed into an herbarium, frozen in an ice sample, and the tun inside it would wait through all of that, requiring nothing, spending nothing, ready. What happens inside the tun is the more interesting part, though it takes a moment to arrive at. The most immediate problem desiccation creates for any living thing is the problem of what water actually does inside a cell. Water is not simply a medium that living systems float in. It is a functional part of the architecture. Cell membranes are held in their characteristic shape, that flexible double layer of phospholipids that controls what passes in and out, partly because of how water molecules arrange themselves around the hydrophilic and hydrophobic portions of the membrane. Proteins fold into the shapes that make them functional because of water's involvement in determining which portions of the protein are exposed to the surrounding liquid and which are tucked inward. Remove the water, and both structures tend to fail. Membranes collapse, fuse, or rupture. Proteins lose their shape and stop working. The interior of a dehydrated cell, without any intervention, tends to become a damaged and disorganized thing. The tardigrade has two separate answers to this problem, and both of them are worth sitting with. The first answer involves a sugar. Many tardigrade species, though not all, produce, as they dry, a disaccharide called trehalose. A disaccharide is simply two sugar molecules joined together, and trehalose is a particular pair of glucose molecules connected in a way that makes the resulting compound unusually stable and unusually useful. As the water leaves the tardigrade's cells, trehalose accumulates around the membranes and proteins that would otherwise be exposed to the damage of drying. It inserts itself into the spaces the water has left. It forms hydrogen bonds with the same sites on those structures that water would have occupied. Holding the membranes and proteins in the right configuration, even in the absence of the liquid they depend on. As the concentration of trehalose increases and the remaining water continues to leave, the trehalose does something else. It solidifies, not into a crystal. Crystals have sharp, regular structures that would press against and damage the cellular components they surrounded, but into something called an amorphous glass, a solidified state without internal order, a material that holds its contents in the same suspended, undamaged arrangement they occupied when the liquid was still present. Think of the difference between freezing a wet sponge and embedding it in clear resin. The frozen sponge holds its shape under the ice's pressure, but remains exposed to ice crystal formation and mechanical stress. The resin-embedded sponge simply stops, held exactly as it was, immobilized without force. The glassy trehalose matrix is something like the resin. The cellular architecture inside it is held in place, not crushed, not reorganized, simply preserved until the water returns to dissolve the glass and restore the liquid environment the cell knows how to function in. Trehalose is not unique to tardigrades, where other organisms use it, certain yeasts, some insects, a number of plants with drought tolerance, the resurrection plant that can dry to a papery crumble and green again after rain. What is particular to tardigrades is not the sugar itself, but the degree to which they deploy it, and the fact that it works alongside a second mechanism that is unique to them. That second mechanism was identified by researchers at the University of Tokyo, and it involves proteins that were not known to exist before the tardigrade was studied carefully enough to look for them. These proteins belong to a class now called CAHS proteins. The initials stand for cytoplasmic abundant heat soluble, which describes their location, their quantity, and their stability under heat, and they do something that no equivalent protein class had been documented doing in any other animal. In the tardigrade's normal, hydrated state, CAHS proteins are dissolved in the cytoplasm. They are present in large quantities, but they are mobile, fluid, participating in the general liquid chemistry of the cell without forming any particular structure. As the cell begins to lose water, as the conditions of desiccation take hold, these proteins change. They begin to polymerize, to assemble into longer and longer chains, and those chains begin to form a gel-like network that spreads through the interior of the cell. As the drying continues, the gel thickens. It transitions from a loose, flexible mesh to something more solid, more fixed, a matrix that physically encases the structures around it, the mitochondria, the ribosomes, the fragments of the cytoskeleton, the membrane systems, all the internal components of the cell that are vulnerable to the mechanical stresses of drying. The comparison that comes naturally is packing material, the foam or paper or formed padding that surrounds something fragile when it needs to travel. The CAHS protein matrix does something structurally similar to what packing material does. It immobilizes what it surrounds, preventing it from shifting, colliding, fragmenting, or fusing with adjacent structures. The cell becomes, in this state, very nearly solid. The normal liquid activity of the cytoplasm, the constant movement of molecules, the flow of vesicles, the sliding of proteins along cytoskeletal tracts stops because there is no longer a liquid medium for it to happen in, and because the solid matrix that has replaced that medium holds everything in place. When water returns, when a raindrop reaches the moss, when the humidity climbs, when the dried specimen is placed in liquid, the CAHS proteins dissolve back into the cytoplasm. The matrix that was solid becomes fluid again. The cellular structures it held are released, intact, into a restored liquid environment. The cell can resume, the tardigrade can resume. What is notable about this system is not just that it works, but that it works reversibly, and that it works at the scale of individual proteins and membranes, the smallest scale at which a cell can fail. The damage that desiccation would normally cause is not repaired after the fact. It is prevented during the process. Because the two mechanisms together, the trehalose glass that holds membranes and proteins in their functional shapes, and the CAHS protein matrix that physically immobilizes the cell's interior, are less like a rescue system and more like a method of careful packaging. The tardigrade does not survive desiccation by recovering from it. It survives by ensuring that nothing requiring recovery actually happens. There is still the question of what the animal's metabolism is doing during all of this. In the ton state, the tardigrade's metabolic rate drops to approximately 1/10,000th of its normal level. That number is not a reduction in the way that a sleeping animal's metabolism is reduced, or the way a hibernating animal's metabolic rate falls through winter. It is a reduction so close to zero that by every standard physiological measurement, the ton is indistinguishable from something that is not alive at all. No detectable oxygen is being consumed. No detectable carbon dioxide is being produced. No electrical activity passes through the nervous system. No nutrients are being processed. The energy budget of the animal, which in its active state would show the constant small expenditures of a living thing maintaining itself, drops to a value that instruments can barely resolve from background noise. What this means for time is interesting to consider. For a metabolically active animal, time passes in the currency of biological processes, heartbeats, cell divisions, the oxidation of sugars, the replacement of worn proteins. An animal that is doing all of these things is aging in a biochemical sense, even when it is still. An animal doing none of them is not aging in any meaningful way. The ton, in this state, is not aging. It is waiting, but waiting in a form that does not wear out by waiting, that does not spend itself in the act of persisting, that is simply there, present in the world without participating in it. A ton recovered from a museum herbarium specimen, a dried piece of moss that had been sealed in a cabinet for decades, can be rehydrated and will sometimes produce a living tardigrade. A ton held in dry conditions at low temperatures can remain viable across timescales that are not easy to think about from the perspective of an animal that ages continuously. The dried moss in a laboratory storage jar is not a grave. It is closer to a waiting room, although that comparison carries more implication than the animal would recognize. The metabolic pause also has consequences for the tardigrade's DNA. Even in the ton state, DNA can sustain damage from residual background radiation, from the slow accumulation of chemical stress, from the unavoidable molecular noise of even the quietest possible existence. Ionizing radiation, in particular, can break DNA strands directly. The tardigrade's DNA, like any organism's DNA, is vulnerable to this. What is less ordinary about the tardigrade is what happens when the animal rehydrates and resumes activity. Tardigrades possess DNA repair systems that are efficient and extensive by the standards of complex multicellular animals. When a rehydrating tardigrade finds that its genome has sustained damage during its time in the ton, double-strand breaks, where both sides of the DNA double helix have been cut through, the most severe form of DNA damage, it can reassemble those breaks in large numbers, simultaneously, and with a high degree of accuracy. Studies exposing tardigrades to doses of ionizing radiation far above what would be lethal to most animals have shown the capacity to repair hundreds of double-strand breaks in a single recovery period, the genome being reconstructed from fragments with a fidelity that allows the animal to resume normal function and reproduction. The repair system is not unique in its basic mechanisms. All organisms have some capacity for DNA repair, but it is notable in its efficiency and its tolerance for large-scale damage. An animal that might accumulate substantial genomic fragmentation over a long period in the tune state does not wake damaged. It wakes, repairs, and continues. The molecular record of whatever it endured while it was still is addressed quietly without any change in the animal's subsequent behavior or biology. There is a protein identified in tardigrade research that has come to be called deposible damage suppressor, which appears to physically associate with DNA and provide some degree of shielding against the strand breaks that ionizing radiation causes. The protein does not prevent radiation from reaching the cell, but it appears to reduce the severity of the damage that radiation produces, possibly by occupying binding sites on the DNA that would otherwise be more vulnerable. When DESUP was introduced into human cultured cells in research settings, those cells sustained less radiation-induced DNA damage than control cells without it, a finding that sits in the background of ongoing research into radiation protection, though its practical applications remain at an early stage. These mechanisms together, the Trello's glass, the CAHS protein matrix, the metabolic arrest, the DNA repair pathways, the DSERP protein, do not exist in the tardigrade as isolated special features. They form something more like a coordinated system, a set of responses that come online together as conditions change, each addressing a different aspect of the problem that extreme desiccation presents. The Trello handles the architectural integrity of membranes and proteins. The CHS proteins handle the mechanical stability of the cell interior. The metabolic arrest reduces the ongoing biological costs to a level that can be sustained without consumption. The DNA repair machinery addresses the damage that persists through all of that and still accumulates. They are not backup systems, they are the system. What the tardigrade does not do in any of this is decide. There is no moment of choosing to enter the ton, no evaluation of conditions, and deliberation about the appropriate response. The whole process is chemical, automatic, driven by the detection of water loss and the cascade of molecular events that detection initiates. The legs draw in because the cells that control them have reached a state that causes them to draw in. The trailose accumulates because the enzymes that produce it are activated by the same signal. The CAHS proteins gel because the drop in water concentration directly drives their polymerization. The animal that emerges from the ton is the same animal that entered it, in the sense that nothing about its identity, its cellular architecture, or its biological program has changed, but the entry and the exit are not things it chose. They are things that happen to it, and then happen to it in reverse. The moss surface where all of this takes place is not a dramatic one. It is usually ordinary. A clump of garden moss on a brick wall, a patch of lichen on a stone, a small accumulation of vegetation in a gutter or a crack. The events that produce a ton are the same events that produce ordinary drying in ordinary weather. A sunny afternoon, a dry wind, the natural fluctuation of humidity that mosses and the animals living in them have been managing for as long as both have existed. The tardigrade does not enter the ton as a last resort. It enters it as a routine matter, the way an animal with gills moves water across them, or the way a plant closes its stomata in dry air. The apparatus is extraordinary by the measures biologists apply to it. The occasions that trigger it are in most of the places tardigrades live, fairly common, in a clump of damp rooftop moss, On a day when rain has fallen and the film of water on each green filament is full and generous, several dozen tardigrades might be moving at any given moment, feeding, negotiating the three-dimensional architecture of the moss, carrying out the small bounded work of their lives. The same clump on a dry afternoon might contain the same animals in the ton state, all movement suspended, all metabolism quieted, the whole population waiting in a form that requires nothing from the world around it and gives nothing back. A day later, if rain falls again, those same animals extend their legs and resume. The transition takes minutes to hours in each direction. Coming out of the ton, the rehydration triggers the dissolution of the CAHS matrix, the re-expansion of cellular membranes, the return of metabolic activity, the unfolding of legs. The whole process of revival for an animal that may have been still for months or years takes less time than it takes a seed to germinate, less time than it takes a butterfly to emerge from a chrysalis. The tardigrade does not wake gradually. It resumes with the specific abruptness of a system restarting, and within the first hour after rehydration, it is walking again, walking at the same pace it always walks, moving with the same rolling, deliberate gait on 8 legs that end in claws or adhesive pads, returning without ceremony to the particular small work of finding food and moving through the water film that makes its world. Whatever the ton contained, whatever duration of stillness, whatever accumulation of damage that has since been repaired, it does not show in the animal's subsequent movement. The resumption is as seamless as the entry. The legs unfold, the animal walks, on a piece of moss somewhere, in a garden, or in a forest, or on a stone wall in a city, or frozen into the surface of an Antarctic clump of vegetation at the edge of a cold sea. A tardigrade that was a dry
drail an hour ago is now moving through the thin film of water that coats a green cell.
Its pace is unhurried. It has the deliberate, compact motion of something that has been moving this way for a very long time, in a body that was built specifically for this, for the water film, for the moss, for the rhythm of wet and dry that governs the only world it has ever needed.
The water around it is generous now, full and clear, filling all the spaces between the moss cells, with the medium the tardigrade moves through most comfortably. The legs work in their slow alternating sequence. The rounded head is forward. The body, which was barrel-shaped and still and patient, is extended again and in motion. If the water retreats tomorrow, it will draw itself inward again with the same unhurried automatic calm. But for now, in the time when the water has returned, the tardigrade moves through the film of it with the same unhurried rolling pace it has always used, resuming as it always does, exactly where it left off. On September 14th, 2007, a rocket lifted from the flat steppe of Baikonur Cosmodrome in Kazakhstan and climbed into a sky that was still, at that hour, a deep and ordinary blue. The Photon M3 spacecraft rode the plume of its Soyuz carrier upward through the atmosphere, past the point where the air thins to almost nothing, and into the particular silence of low Earth orbit. Mounted on the exterior of that spacecraft, bolted to its outer surface in a compact cylindrical housing called Biopan 6, was a biological exposure platform. And inside that platform, sealed in their specimen carriers, were tardigrades, dried, curled, and entirely still, in the compact barrel form their bodies take when water is gone and the world has asked too much of them. They had no awareness of the trajectory they were on. That is not a poetic observation, only a factual one. In their desiccated state, with metabolic activity reduced to a trace so faint as to be functionally undetectable, There was nothing inside those small bodies registering velocity or altitude or the growing distance between them and the surface of the planet they had always lived on. The rocket climbed, the spacecraft reached orbit, the Earth curved away below, and the tardigrades in their sealed carriers remained as they had been, still, dry, and waiting. Photon M3 was an unmanned mission operated by the European Space Agency, part of a long series of automated spacecraft designed to carry experiments into orbit and return them to Earth for analysis. It was not built for the tardigrades specifically. It carried a range of biological and physical experiments across its interior and exterior platforms, the kind of broad scientific payload that makes use of every available surface when a spacecraft is going anyway, and the cost of each kilogram has already been paid. The Biopan 6 platform was one of those surfaces, a biological exposure unit designed to open its protective cover once in orbit, allowing whatever was placed inside to experience the space environment directly, with no atmospheric buffer and no pressurized module between the specimens and the vacuum outside. The cover opened, and the specimens were met by a set of conditions that do not exist anywhere on the surface of Earth. The vacuum of low Earth orbit registers at around 10 to the power of -7 Pascal. To give that figure some physical shape, standard atmospheric pressure at sea level is approximately 101,325 Pascal. The pressure at the top of Everest is around 33,700 Pascal. At the edge of the stratosphere, where commercial aviation gives way to the emptiness above, pressure has fallen to something in the low hundreds. The pressure that met the tardigrades when the Biopan 6 cover opened was so far below any of those numbers that the ordinary scale of terrestrial measurement stops being useful. It was, in the most direct sense of the phrase, nothing, or as close to nothing as the natural world produces at accessible altitudes. Before the mission, no animal had been confirmed to survive direct exposure to that kind of vacuum outside of a sealed container or pressurized suit or pressurized module of any kind. Microorganisms had been tested in simulated space conditions. Bacterial spores had shown survival under various experimental approximations, but a multicellular animal, an organism with differentiated tissues, a defined body structure, internal organs, legs, and a cuticle that separates its internal environment from the world outside, had not previously been placed directly in open vacuum and recovered alive. Photon M3 would be the first confirmation that such a thing was possible. The two species selected for the mission had been chosen with care. Rictersius coronifer was originally collected from mosses growing on rock faces in Scandinavia, gray-green clumps clinging to stone in the northern light, the kind of habitat where drying and rehydration happen repeatedly through the year as the weather moves through its cycles. Rictersius coronifer had been studied for its confirmed ability to withstand repeated severe dehydration and return to normal activity without apparent degradation. It was a known quantity, experimentally speaking, with a documented tolerance for the disappearance of water. The second species was Milnesium tardigradum, a predatory tardigrade that feeds on other small invertebrates and single-celled organisms, and which had shown, in prior laboratory work, a particular robustness that made it a useful candidate for exposure experiments at the far end of what was likely survivable. Both species were dried before the mission, entering the compact tun state in which their bodies would remain for the duration of the flight. In that state, they were placed into their specimen carriers and mounted in the Biopan 6 platform. Some carriers were arranged to expose the tardigrades to the full range of the solar spectrum, including ultraviolet radiation at wavelengths the Earth's atmosphere ordinarily absorbs before they can reach the surface. Others were partially shielded, allowing exposure to vacuum and the broader radiation environment of orbit while filtering out the most energetically intense ultraviolet wavelengths. The distinction between those two conditions would matter considerably when the results came back. Sunlight in low Earth orbit is not the sunlight that reaches the ground. The atmosphere filters out a substantial portion of the solar spectrum before it arrives at the surface, particularly in the ultraviolet range. UVC radiation, which carries enough energy to break the chemical bonds in DNA directly, is almost entirely absorbed by the ozone layer. UVB, which causes sunburn and DNA damage at the surface, is partially reduced. UVA passes more freely. In orbit, without that atmospheric buffer, all three arrive at full intensity. The total ultraviolet dose received by the unshielded tardigrade specimens over the course of the mission across UVA, UVB, and UVC combined came to approximately 7,000 kilojoules per square meter. That figure represents a UV exposure orders of magnitude beyond what would be lethal to human skin or to the DNA of most organisms adapted to life beneath an atmosphere. The spacecraft completed its orbits, and 10 days passed, the Earth turning below at a pace that meant the spacecraft crossed it roughly 16 times every 24 hours, moving through alternating arcs of sunlight and the cool darkness of the planet's shadow. The tardigrades and their carriers received the vacuum and the radiation and the temperature swings between sun-facing and shadow-facing positions, none of which registered in any biological process within their still bodies, because biological process had for the duration of the journey essentially paused. On September 26, 2007, Fedonem 3 re-entered the atmosphere and landed by parachute on the Kazakh steppe. Recovery teams retrieved the spacecraft and removed its experimental payloads. The Biopan 6 platform was transferred to the team that had designed the tardigrade experiment, and the specimen carriers were transported to a laboratory where the analysis would begin. The question at that point was straightforward. Even if the answer was not yet known, would any of the animals that had been in open space for 10 days, in vacuum, and in some cases under unfiltered solar ultraviolet radiation of significant total dose, resume normal biological function when water was reintroduced? The rehydration process in a tardigrade experiment is not a dramatic procedure. A technician places the specimen carrier under magnification, adds water to the dried material containing the animals, and then waits. The response, when it comes, is slow. Legs that have been retracted and held in the curled ton form begin, over the course of minutes, to extend. The body, which in the dried state has the appearance of a small sealed pellet, begins to resume its rounded but more elongated living shape. Movement, when it starts, is tentative at first, a slight shifting of position, a slow extension of one limb, then another. It is not a sudden return. It is a gradual re-engagement with the physical world, the kind of resumption that happens in its own time and cannot be hurried. Among the specimens that had been shielded from the full solar ultraviolet spectrum, exposed to vacuum and the broader radiation environment of orbit, but not to the unfiltered UVC and UVB wavelengths, survival rates after rehydration were comparable to those of ground control specimens that had been dried and kept on Earth for the same period. The animals moved, they fed. In the weeks following rehydration, they produced eggs. Those eggs were viable. The animals that hatched from them developed normally, completing their life cycle without visible disruption attributable to the flight. Among the specimens that had received the full, unfiltered solar ultraviolet dose, the 7,000 kilojoule per square meter exposure across all UV wavelengths, survival was much lower, as the researchers had broadly anticipated. A UV dose of that magnitude carries enough energy to cause widespread damage to DNA and cellular structures, and the results reflected that. The large majority of specimens exposed to the full solar UV spectrum did not revive. That outcome, given the dose involved, was not a surprise to the team. What drew more attention was that a small percentage of specimens from the full UV exposure condition did revive. They were fewer than in the shielded group. Their survival rate was not high, but some of them moved, and some of them reproduced, and the eggs some of them laid were viable. The total UV dose they had received represented a level of radiation exposure that should, by any prior expectation, based on the sensitivity of biological molecules to that kind of energy, have been uniformly fatal. For most of the specimens, it was. For a small number, it was not. The researchers noted this carefully and recorded it precisely, as one would record any experimental result that does not fully conform to what the available model predicts, without overstating what the finding meant and without understating the fact that it was, within the controlled terms of the experiment, a real and documented result. Richtersius coronifer, the species originally collected from Scandinavian mosses, performed consistently across both UV conditions, present in both the group that survived at high rates and the group that showed only partial survival. Its post-flight behavior documented in detail in the weeks following the mission's return. Milnesium tardigradum, the predatory species, showed what the post-flight survival assessments would eventually characterize as particularly notable resilience, with performance in the recovery phase that distinguished it among the species tested. The egg-laying in the weeks after the flight is worth dwelling on for a moment, not because it is the most technically significant finding, but because it is the most concrete image of what the experiment ultimately showed. A tardigrade that returned from 10 days in open space, in vacuum, exposed to an orbital radiation environment, was not merely alive in a minimal, barely functional sense. It was alive in the way that animals are fully alive, carrying out the complete biological cycle that defines the continuation of a species. The eggs it laid were not artifacts of a compromised system producing damaged structures. They were small, intact, developing normally, hatching into animals that would themselves grow and feed, and in time produce eggs of their own. The Biopan 6 platform, after the experiment concluded, was processed and archived. The data from the mission was published in the journal Current Biology in 2008 by a team led by K. Ingemar Jönsson and colleagues from Kristianstad University in Sweden, along with collaborators from other European institutions. The paper reported, in the measured language of peer-reviewed biology, that tardigrades had survived direct exposure to the space environment, including open vacuum and partial or full solar ultraviolet radiation, and that a subset of survivors had reproduced successfully after return. It was, by any reasonable interpretation, a documented first, and it was documented as such, carefully, with the appropriate qualifications about exposure conditions and survival rates, and the specific distinction between the shielded and unshielded groups, without the language of the paper, suggesting that what had happened was anything other than the result of a well-designed experiment conducted under controlled conditions, observed carefully, and reported precisely. The context that makes the Photon M3 result legible is the biology that the tardigrades brought with them into orbit. They did not develop any new capacity for the mission. They did not acquire, in the weeks before launch, some additional resistance specific to the space environment. What they carried was what they had always had, a set of biological mechanisms evolved over a very long time in the context of a planet where conditions at the small scale, the scale of a moss film, a lichen surface, a patch of soil between stones, can change dramatically and rapidly, where water appears and disappears, where exposure to UV radiation on a desiccated surface without the protection of surrounding water is a real condition that tardigrades encounter in ordinary terrestrial life. The tune state in which they made the journey is the same state that a tardigrade enters when a rooftop moss dries out on a warm afternoon. The DNA repair capacity that allowed some specimens to survive even the full UV exposure is the same capacity that tardigrades employ in their ordinary habitat. None of it was exceptional in the sense of being specific to space. It was exceptional in the sense of being, relative to most other animal biology, very highly developed. The Photon M3 result did not, in the months and years after its publication, settle all questions about tardigrade resistance to the space environment. It opened several questions about whether longer durations of exposure would produce different outcomes, questions about what role the specific UV wavelengths played relative to the vacuum exposure alone, and whether those effects could be further separated under different experimental designs, questions about the mechanisms underlying the partial survival of specimens in the full UV condition, whether some individuals carried greater amounts of protective proteins, whether the geometry of their position in the carrier provided any partial shielding, whether there was biological variation within the species population that corresponded to the difference between survivors and non-survivors. Those questions were not answered by the 2007 mission, and some of them remain open. What the mission established was the documented, peer-reviewed, replicated by analysis fact that tardigrades in the tone state can survive 10 days in open space, and that a portion of those survivors can reproduce normally after return to Earth. That is what the data showed. That is what was reported. The questions that followed from it are the normal scientific response to a result that extends what was previously understood. The BioPan platform itself had been used in earlier missions to expose other biological material to the space environment and would be used in subsequent experiments as well. Tardigrades were not the only organisms aboard Photon M3. The spacecraft carried bacteria, plant seeds, and various biological samples as part of its broader experimental manifest. The tardigrade result was the one that carried the specific distinction of representing the first multicellular animal to be confirmed as a space environment survivor in the direct exposure sense, outside any protective enclosure, in the vacuum itself. In the weeks after the mission's data was fully processed and the survival assessments were complete, the laboratory dishes containing the rehydrated post-flight specimens held a small collection of living animals. Some were from the shielded group, moving through the water on their sample slides in the same unhurried way that tardigrades have always moved. Some were from the unshielded group, fewer in number, but present and reproducing, and distributed across several of those dishes in the days that followed were small clusters of eggs. Tardigrade eggs are difficult to see without magnification, being roughly the size of a tardigrade itself, which is to say the size of a period at the end of a sentence, or smaller. Under a microscope, the eggs of Rictersius coronifer are visible as small, roughly spherical structures, sometimes with surface ornamentation that varies by species, laid singly or in small groups depending on the reproductive method the species uses. The eggs laid in the post-flight dishes were intact. They developed. The animals that hatched from them were normal, the same body plan, the same 8 legs, the same rolling gait that had been described by the naturalists who first noticed tardigrades moving through drops of water centuries before any spacecraft existed. There is something specific about that image, the post-flight egg, developing normally in a laboratory dish, that carries the weight of what the experiment actually showed more fully than any of the numerical results. The vacuum exposure figure, the UV dose figure, the survival rate figures, these are real and important and are what the science rests on, but the egg is the concrete endpoint of what those figures describe. An animal that had been in open space, in vacuum, under radiation, returned to Earth, was rehydrated, lived, and reproduced. The next generation was present, in that dish, developing toward the same small, unhurried life that tardigrades have been living in. Moss films and lichen surfaces and soil samples for as long as the genus has existed. The Baikonur Steppe, where the spacecraft launched, is a flat, semi-arid landscape in central Kazakhstan, a place where summer temperatures can be extreme, and the soil is often dry and sparse with vegetation. Whether tardigrades live in the thin mosses and lichens that grow there is not something the mission report specified, but the habitat type is one they are commonly found in. The same animal that rode the rocket from that steppe into orbit, in its dried and curled form, was in all likelihood also present in the soil and moss of the launch site itself, in populations that had no awareness of the spacecraft above them, moving through their water films in the ordinary way. That is not a detail the researchers included in their paper because it was not relevant to the experiment. It is simply the character of the animal being studied, widespread enough and distributed into enough different habitat types on enough different continents that the place from which a spacecraft departs to carry tardigrades into orbit is probably also a place where tardigrades already live. The mission's results have been referenced in the years since, in research papers dealing with astrobiology, the study of life's potential distribution and survival beyond Earth, and in papers dealing with the practical biology of tardigrade resistance. They appear in discussions of panspermia, the hypothesis that biological material might travel between planetary bodies as an example of the conditions that such material would need to survive. They appear in discussions of space biology and the limits of multicellular animal tolerance. They appear in papers specifically about tardigrade extremophile properties, placed alongside the laboratory results from pressure experiments and temperature experiments and radiation exposure experiments as one data point in a wider picture of what this particular animal can survive. The Photon M3 result is, in that broader context, one mission among several that have studied tardigrades in or near the space environment. It was the first, and it remains the clearest single demonstration that the question of whether a multicellular animal can survive direct exposure to the vacuum of space has a documented answer, arrived at through a controlled experiment, carried out under real orbital conditions, with a result that has been formally published, peer-reviewed, and absorbed into the ongoing scientific literature on both tardigrade biology and the conditions of the space environment. The spacecraft that carried them has long since been recovered and processed. Its instruments were removed, its experimental payloads were distributed to the research teams that had designed them, and the vehicle itself was retired to whatever archival status unmanned spacecraft acquire after their mission is complete. The tardigrades that flew on it, the ones that survived, the ones that reproduced, have by now lived out their lives in the laboratory dishes, where they were rehydrated. Their descendants may or may not still exist in some form in some collection. Tardigrades in laboratory culture can be maintained, but culture maintenance requires ongoing attention, and research priorities shift. What persists is the record. The paper published in Current Biology in 2008, the survival data, the UV dose measurements, the post-flight reproductive success figures, the eggs somewhere in the weeks after Photon M3 returned from orbit, in a laboratory that smelled of the particular mild chemical scent of biological research facilities. A researcher placed a sample carrier under magnification and added water to dried tardigrades that had been in open space 10 days earlier. The room was probably quiet. The microscope light was on. The researcher watched the field of view and waited to see whether anything would move. Something did. It moved the way tardigrades always move, slowly, with the rolling eight-legged gait that is the characteristic motion of the phylum, the same pace that has been observed since a naturalist in the 18th century first pulled a clump of dried moss apart and looked at what was living in it. The legs extended, one after another. The body settled into its living, elongated form. The animal moved across the surface of the slide. In the weeks that followed, in the dishes where the surviving post-flight specimens were kept, eggs appeared. Small, intact, developing normally, carrying inside them the biological information for the same basic body plan: 8 stubby legs, a round head, a cuticle, a slow and deliberate way of moving through whatever thin film of water the world provides, that the parent had carried into orbit and back again. The eggs rested in their dishes under the low warmth of the laboratory. The water around them was still. The animals inside them were developing in the quiet, unhurried way that tardigrades develop, becoming in their own time the next version of something that has been living in this patient, persistent, water-seeking way since long before any human thought to look for it, long before any moss was examined under any microscope, long before any spacecraft was built to carry anything beyond the edge of the atmosphere and bring it home again. The eggs held, the water was still, and in that stillness, in that small and ordinary dish, the next generation was already forming. In a laboratory measurement, the unit used to describe radiation absorbed by living tissue is called a gray. 1 gray is the absorption of 1 joule of energy per kilogram of mass. At 4 gray, delivered to the whole body, a human being faces serious and likely fatal damage to the cells lining the gut and the bone marrow tissue that produces blood. At 8 gray, survival without intensive medical intervention becomes unlikely. These are not large numbers in physical terms. In the context of what the body can manage, they mark a boundary that is relatively close to the surface of what ordinary cellular repair can handle. In a separate room of the same kind of laboratory, a tardigrade inside an exposure chamber receives a measured dose of ionizing radiation from a controlled source. The dose is calibrated in the same units, the same gray, the same measure of energy delivered per kilogram of mass. The tardigrade is in its active walking state. Water surrounds it. Its legs are extended. Its metabolic processes are proceeding at their normal rate. The dose climbs. It passes the point at which a human cell population collapses. It continues past the point at which virtually all other animal tissue would sustain irreversible damage. At around 570 gray, in its active state, the tardigrade is still viable. The cellular repair processes inside it continue to function. When the exposure ends and the animal is returned to normal conditions, it resumes its slow walk across the slide. The gap between 4 gray and 570 gray is not a small one. It represents a difference of more than 100-fold. And that figure, 570 gray, in the active hydrated state, is not where the tolerance ends. When the same tardigrade is desiccated, curled into the compact barrel-shaped resting form its body produces under dry conditions, the threshold climbs further still. In its desiccated state, a tardigrade can survive doses approaching 6,200 gray. The cellular architecture that remains viable through that kind of exposure, the DNA repair systems that reassemble fragmented genetic material at that scale, represents something that falls near the outer edge of what has been documented in any complex animal. The comparison that appears most often in the scientific literature involves a bacterium rather than an animal. Deinococcus radiodurans is a single-celled organism with a tolerance for ionizing radiation that has been studied in detail for several decades. It can survive doses of approximately 5,000 gray. Its resistance comes from a combination of extraordinary DNA repair enzymes and the maintenance of multiple redundant copies of its genome, so that when radiation shatters one strand of genetic material, other copies remain intact and serve as templates for reconstruction. For a single cell, this is a deeply effective strategy. Deinococcus radiodurans has appeared on lists of the most radiation-resistant organisms on Earth in research papers and reference texts alike, and the designation is reasonable given what it can endure. The point that tardigrade researchers return to is not that the tardigrade exceeds this figure by a large margin in its active state. In its active state, it does not. The point is what the tardigrade is. Deinococcus radiodurans is a single cell. It has no tissues. It has no organs. It has no nervous system, no digestive tract, no musculature, no sensory structures. When it repairs its DNA after extreme radiation exposure, it is performing that repair within a single bounded cell membrane, working on a genome shared across identical copies. The tardigrade, at its radiation tolerance, is doing something structurally different. It has differentiated tissues. It has a body with distinct regions that serve distinct functions. It has muscle cells that move its 8 legs, cells lining a pharynx that grinds its food, cells forming a thin but real nervous system running along its body. All of these different tissue types sustain radiation damage, and the animal's biological systems repair that damage in a coordinated way across a body that has actual structural complexity. That distinction matters because multicellular organisms with differentiated tissues generally do not achieve radiation tolerances approaching those of single-celled organisms with redundant genomes. The cellular repair mechanisms available to complex animals are in most cases constrained by the need to maintain the integrity of many different cell types simultaneously, each with its own function and its own vulnerability. The tardigrade achieves a radiation tolerance that sits close to Deinococcus radiodurans, while also being an animal in the full sense, segmented, legged, capable of feeding, of reproducing, of responding to its environment through sensory structures. This is the quality that tends to occupy researchers who study it, not the number itself, but the kind of organism the number belongs to. At temperatures far below what any ordinary biological process can manage, a different kind of measurement applies. The Kelvin scale places absolute zero at the point where molecular thermal motion ceases entirely. 0 Kelvin is minus 273, 15 degrees Celsius. At temperatures approaching that point, chemical reactions that sustain cellular life do not slow. They stop. Enzymes cannot function. Membranes lose the flexibility needed to maintain their structure. Water, the medium in which all known cellular biochemistry occurs, is not present as a liquid. It is absent or frozen into a rigid lattice, or in the case of conditions approaching absolute zero, reduced to a state where the concept of liquid has no meaningful application. A desiccated tardigrade cooled in a controlled laboratory setting to within 1 degree of absolute zero to minus 272 degrees Celsius has been brought back to normal activity upon return to ambient temperature and gradual rehydration. The animal rests at that temperature in a stillness that is, by most available measures, complete. Its cellular structures are held in a dry glass-like matrix that formed as the water left its body. The molecular motion within it is reduced to nearly nothing. Time, in the biological sense of processes occurring and accumulating, has effectively stopped. When warmth and water return, those processes resume. The tardigrade moves again, at the opposite end of the thermal scale, briefly exposing a desiccated tardigrade to temperatures above 150 degrees Celsius, temperatures that exceed the boiling point of water by 50 degrees, temperatures approaching those of a domestic oven set to its highest setting, has in laboratory conditions produced animals that survived and became active again after rehydration. These are not sustained exposures at these temperatures. The duration matters, and the conditions under which the animal enters this state matter. But the range between roughly minus 272 and plus 150 degrees Celsius represents a thermal survival window, that is, across documented animal life, without a close comparison. The same body form, 8 legs, a rounded head, a cuticle that is shed and replaced as the animal grows, that occupies a thin film of water on a piece of rooftop moss, also carries within it the biological capacity to remain viable through that span of conditions, not because each individual encounters all of those conditions over a lifetime. Most tardigrades live through ordinary fluctuations of temperature and moisture without approaching any of these extremes. The tolerance is present as a biological fact without being routinely exercised, the way a structure can have a load rating far above what it ever bears in normal use. The question of pressure tolerance follows a similar pattern. The deepest point of the Mariana Trench in the Pacific Ocean sits at approximately 11,000 meters below the surface. At that depth, the weight of the water above produces a pressure of roughly 1,086 atmospheres, more than 1,000 times the pressure at sea level. Submersibles designed to reach that depth require careful engineering to keep their interiors intact. Organisms that live at those depths have evolved body structures and biochemistries adapted to function under that sustained compression. For most animals from the surface, a fraction of that pressure would be lethal. In a laboratory setting, tardigrades suspended in water have been subjected to hydrostatic pressure using sealed chambers and hydraulic presses, with the pressure raised incrementally to levels far beyond what the deepest ocean trench produces. Pressures of approximately 6,000 atmospheres have been reached in these experiments, roughly 5.5 times the pressure at the floor of the Mariana Trench. The tardigrades were not destroyed. When the pressure was released and the animals were returned to normal conditions, at least a portion of the specimens remained active. The results have varied across different experimental designs, and not every individual in every test survives every exposure. But the survival of some portion of a tardigrade population at 6,000 atmospheres places the animal's pressure tolerance well above any ocean floor currently accessible to study. The specific reason tardigrades tolerate high hydrostatic pressure without the cellular disruption that would affect most other animals is connected in part to what happens inside their cells under those conditions. The proteins and cellular membranes of organisms adapted to high pressure generally have structural features that allow them to remain functional under compression, a flexibility in their molecular geometry that prevents them from becoming rigid or losing their shape when surrounded by enormously compressed water. Tardigrades appear to share some of these properties, though the exact molecular mechanisms of their pressure tolerance are an area of continuing research rather than fully settled science. What is clear from experimental results is that the outcome of high pressure exposure, survival, and subsequent normal activity has been replicated across different studies using different species with consistent results. Each of these categories of extreme condition, temperature, radiation, pressure, represents a distinct physical challenge to biological tissue. Low temperature interferes with the chemical kinetics that sustain cellular processes. High temperature disrupts protein folding and membrane integrity. Ionizing radiation damages nucleic acids and produces reactive chemical species that destroy cellular components. High pressure compresses molecular structures and alters the behavior of proteins and lipids in ways that disrupt their normal function. Desiccation removes the water that serves as the medium and participant in essentially all biochemical reactions. Vacuum, the absence of atmospheric pressure entirely, combines the effects of desiccation with exposure to ultraviolet radiation and the near-zero temperature of open space. These are not variations on a single stress. They are different kinds of damage, operating through different mechanisms, requiring different biological responses. An organism that demonstrates documented survival across all 6 of these categories occupies a position that biologists have given a specific name. The word extremophile refers to any organism that thrives in or tolerates at least one category of physical or chemical conditions outside the range that most known life tolerates. Heat-tolerant organisms are thermophiles. Cold-tolerant organisms are psychrophiles. Organisms that survive high salt concentrations are halophiles. The prefix varies. The category is the same, a form of life whose tolerance extends into territory where most others cannot persist. The word polyextremophile combines that root with the Greek for many. A polyextremophile is an organism whose tolerance extends across multiple, distinct categories of extreme condition at once. The designation requires not just broad tolerance in a general sense, but specifically documented survival in physically distinct, separately tested categories. The tardigrade qualifies in at least 6: desiccation, ionizing radiation, extreme temperature at both ends of the scale, extreme hydrostatic pressure, exposure to the vacuum of open space, and in some species, high salinity. Each of those 6 categories corresponds to a real body of laboratory work. The desiccation tolerance has been replicated across dozens of studies in many countries, using many species of tardigrade under varying controlled conditions. The radiation tolerance figures come from carefully calibrated exposure experiments in which doses were measured, animals were monitored, and results were assessed against ground controls. The temperature range has been established through cryogenic cooling experiments and high-temperature exposure experiments using desiccated specimens under controlled conditions, with results confirmed by different research groups working independently. The pressure results come from laboratory chambers designed to maintain precise hydraulic conditions, with survival assessed upon return to ambient pressure. The vacuum and space exposure data come from real orbital experiments, with mission parameters documented by the space agencies involved and results submitted for peer review. The salinity results, which apply to a subset of species rather than all tardigrades, come from aquatic and marine habitat studies, combined with controlled laboratory salinity exposure. 6 categories, 6 corresponding bodies of experimental evidence, the same small animal. What the polyextremophile designation does not mean is that any individual tardigrade encounters all of these conditions in its lifetime, or that surviving them produces no cost. Survival, even when it occurs, is not uniform across every individual in a population exposed to a given extreme. In radiation experiments, some animals survive and some do not. The dose and duration matter, the species matters. Not all of the roughly 1,300 described species have equivalent tolerances across every category. The state the animal is in when exposure occurs matters enormously. An active hydrated tardigrade and a desiccated tun-state tardigrade exposed to the same radiation dose will show very different survival rates, with the desiccated form surviving far higher doses. The label polyextremophile is a classification, not a guarantee of invulnerability. It reflects the empirical finding that this animal's tolerance across a list of physically distinct categories has been measured at levels that no other known complex animal matches in the same breath. Not in any single category necessarily. Other organisms survive comparable or greater extremes in their particular specialty. Deinococcus radiodurans, in radiation tolerance specifically, does not fall far below the desiccated tardigrades figures, and with its redundant genome strategy, it has its own elegance. Certain archaea survive temperatures above 120 degrees Celsius in hydrothermal vents. Organisms living in deep-sea sediments tolerate pressures in the range of thousands of atmospheres, but these organisms are specialists. Their tolerance is, in most cases, narrow. They thrive in one extreme and require it. They are not adapted to the full range. The tardigrade's position is different. Its tolerance is wide rather than deep in any single dimension. It does not require any of these extreme conditions. It is not found only in deep ocean trenches, or only in thermal vents, or only in irradiated environments. It lives, primarily, in the thin film of water, on terrestrial mosses and lichens, on rooftops and garden walls and forest floors, in places that are by no objective measure extreme at all. The tolerance is available to it without being demanded by its ordinary environment. That quality, a wide tolerance carried through an ordinary habitat, is part of what makes the tardigrade's biology a persistent subject of interest for researchers working on questions that extend beyond the animal itself. Work on the proteins that protect tardigrade cells during desiccation has prompted researchers to ask whether those proteins, or structural analogs of them, could be used to stabilize biological materials that are currently difficult to preserve without refrigeration. Vaccines, for instance, require cold storage chains that are difficult to maintain in remote or resource-limited settings. Some biological materials degrade rapidly when removed from controlled temperature environments. The molecular scaffolding that allows tardigrade cells to survive in a dry, stable state without damage has attracted interest as a possible model for preservation approaches that do not depend on cold storage. This research is early stage and ongoing. It has not produced wide practical applications yet, but the direction of the inquiry follows directly from the biology. Research on tardigrade DNA repair mechanisms has similarly attracted interest from researchers working on understanding the molecular basis of radiation resistance more broadly. The specific repair pathways that allow a tardigrade to reassemble hundreds of double-strand DNA breaks simultaneously are not identical to those in other radiation-resistant organisms. Understanding how those pathways work at the molecular level, which enzymes are involved, how they are recruited to damage sites, in what sequence the repair proceeds, is a line of investigation that produces knowledge applicable beyond the tardigrade itself. To the broader question of how DNA repair can be made faster or more reliable under challenging conditions. There is a protein identified in tardigrades, produced in their nuclei, and associated particularly closely with their radiation resistance that has attracted its own line of research. It binds to DNA and appears to act as a physical shield against radiation-induced damage, reducing the number of breaks that occur in the first place, rather than relying entirely on repair after the fact. The finding that an animal carries a protein specifically adapted to protect its DNA from radiation damage suggests that this tolerance is not simply an accidental byproduct of the desiccation adaptations. It has its own dedicated molecular architecture. Understanding what that protein does and how it does it is an ongoing project in several research groups. None of this places the tardigrade in a category that makes it untouchable or infinitely resilient. It can be killed. Sustained exposure to conditions it cannot manage in any form will end an individual animal. There are chemical toxins, specific pathogens, conditions of prolonged heat in its active state, and sustained radiation doses that exceed even its considerable ceiling. The 6-200 gray figure for desiccated state radiation tolerance is a survival threshold for some portion of a population, not a guarantee of total population survival at that dose. The thermal range represents brief exposures at extreme temperatures in dry conditions, not sustained residence in those conditions. The animal is robust in the specific documented ways that experimental biology has established, and within the limits that the same experimental biology has identified. What is useful about the polyextremophile designation held at the level of a simple list is the breadth of the list itself. 6 categories: desiccation, ionizing radiation, extreme cold, extreme heat, hydrostatic pressure, well above the deepest known ocean point, the vacuum of open space, each one a distinct physical challenge requiring a distinct biological response, each one tested, documented, and submitted to review. The same animal, smaller than the period at the end of a sentence when fully extended, carrying 8 stubby legs and a small round head, present in all 6. The stillness of that list, when read at this pace and at this hour, has a particular quality, not a list of feats, a list of conditions. Physical facts about the world that most living things cannot endure, and one animal that has been shown, carefully and repeatedly, to manage across all of them in ways that researchers from many different institutions, working with many different experimental designs, have consistently confirmed. The confirmation is what makes the list meaningful, not the surprise of it, though the gap between the ordinary habitat and the extraordinary tolerance is real, but the cumulative weight of the evidence. A pressure chamber the size of a fist, filled with water and sealed under hydraulic force, a cryogenic vessel cooled within 1 degree of absolute zero. A radiation exposure chamber calibrated to deliver a measured dose with precision. A vacuum chamber from which all but a trace of atmospheric gas has been withdrawn. In each of these settings, at different times by different researchers, the same small animal was placed, observed, and found afterward, in some portion of its population, in the characteristic way biology deals, in percentages rather than certainties, to have remained viable. That survival is not drama, it is data. It is the accumulated output of experiments designed to answer specific questions about the limits of biological tolerance, conducted carefully, with controls, with documented methods, with results that have been replicated or challenged, and often both. The tardigrade is interesting to researchers not because it is impervious, but because it is instructive. Every tolerance it displays is the product of a specific molecular mechanism. Every mechanism is a solved problem in biology, a way that evolution, across however many million years, arrived at a working answer to a physical challenge that would otherwise be lethal. The answers are legible. to some degree in the proteins and repair pathways and structural properties that can now be studied in detail. The table of tolerances, laid out in a reference paper, 570 gray active, 6,200 gray desiccated, minus 272 Celsius, plus 150 Celsius, 6,000 atmospheres, 10 to the power of minus 7 Pascal, reads quietly, each number is a boundary. Each boundary is a point where the biology holds when almost nothing else does, and each of those points was found not by expecting it, but by measuring it, by setting a condition, introducing the animal, waiting, and recording what happened. The 6-category list that earns the polyextremophile designation does not collapse into a single explanation. There is no one thing the tardigrade does that accounts for all of it. The desiccation tolerance involves the glass-like sugar matrix and the gel-forming proteins that protect cellular architecture when water is absent. The radiation tolerance involves the dedicated DNA repair pathways and the shielding proteins that reduce damage before it accumulates. The temperature tolerance at low end involves the stability of dry molecular structures at near 0 Kelvin. The pressure tolerance involves protein and membrane properties that remain functional under compression. These are related in the sense that the desiccated state underlies several of them. The ton state, by removing water and immobilizing cellular components, reduces the damage that radiation, pressure, and temperature extremes would otherwise cause, but the mechanisms are distinct, and they compound rather than duplicate one another. A researcher reading across the literature on tardigrade extremophily is reading across a range of subdisciplines: molecular biology, biophysics, evolutionary biology, astrobiology, radiation science, cryobiology. The animal appears in all of them for different reasons, studied with different tools, generating different kinds of data that nonetheless converge on the same body. That convergence is part of what makes the scientific interest in tardigrades durable. It is not a single striking finding that attracted attention and then faded. It is a persistent relevance across multiple lines of inquiry, each of which keeps returning to the same question: what is this animal doing at the molecular level that allows it to be here, measurably intact after this? The question has partial answers now. The trehalose, the CAHS proteins, the DIS protein that shields DNA, the highly efficient repair pathways, partial answers that are real answers, grounded in experimental evidence that have changed what is understood about the molecular options available to biological systems under stress. The full picture is still assembling. New species continue to be formally described, and not all species have been tested across all categories. The molecular mechanisms identified in the most studied species may or may not apply in the same form to species studied less thoroughly. The research is ongoing, unfinished in the way that most productive scientific inquiry is unfinished, not stalled, but continuing to find more to examine than it started with. Somewhere in that ongoing work, a graduate student opens a labeled vial of tardigrades collected from a piece of Antarctic moss, places them under a microscope, and begins a protocol designed to measure their tolerance to a specific dose of ultraviolet radiation. The animals in the vial are small and for now active. They are moving at their characteristically slow pace across the wet surface of the slide. The experiment will take several days. The results will be added to a growing table of figures, each one representing a boundary that biology has documented, each one belonging to the same small body that was first noticed in a drop of water wrung from dried moss by a German pastor with a primitive microscope in 1773. The same basic form, the same 8 legs, the same rounded head, the same slow rolling gait, and a tolerance spread across 6 distinct categories of physical extreme that is described in the scientific literature with the same flat, careful language used to describe the weight of a stone or the temperature of water, because that is what it is, a measured fact, a documented threshold, a number in a table that belongs to an animal smaller than the period that ends this sentence, moving unhurried and intact through the thin film of water it is always called home. 6 categories, 6 sets of results, the same small animal held calmly in all of them, continuing, in a piece of amber the color of dark honey, in a university collection somewhere in the northern part of the world, there is a tardigrade that has not moved in 99 million years. The resin that holds it is translucent enough that under a strong lens, the claws are still visible, 8 of them, arranged in the same pattern they occupy on every living tardigrade, walking through a film of water on a garden wall right now. A researcher in 2021 described the specimen carefully, comparing those ancient claws under magnification to the claws on a freshly collected modern specimen, and found the structure consistent enough to classify it to genus without difficulty. The amber came from what is now Myanmar. The forest that produced the resin was a Cretaceous forest, warm and wet, and the animal that wandered into it was small enough that the droplet of resin simply enclosed it whole, and the whole thing became stone over the course of time. Too slow to imagine. What that researcher was doing in the act of that comparison was confirming that the tardigrade body plan has held its shape across 99 million years of geological change, not approximately the same shape, the same claws, the same body segmentation, the same basic proportions of a rounded head, a tapered trunk, 8 legs arranged in pairs, the specimen preserved in Cretaceous amber, and the specimen alive on a piece of damp moss in someone's garden right now would be recognizable to the same eye as members of the same kind of animal, a continuity that puts most of evolutionary history into a certain kind of perspective, a perspective that is very quiet and very wide. The amber specimen was named Paradoriforibius chronocaribius when it was formally described, and its classification confirmed it within a lineage, the tardigrade phylum, that researchers using molecular methods have placed much deeper in time than 99 million years. Molecular clock analyses, which estimate the age of lineages by measuring the rate at which genetic sequences diverge, suggest that tardigrades first appeared as a distinct group somewhere between 500 and 540 million years ago. That places their origin in the Cambrian period, during the stretch of geological time when most of the major animal body plans on Earth came into existence within a relatively compressed interval. The world at that point was genuinely different, the continents arranged in configurations that no modern map would help orient a person, the atmosphere measurably unlike the one that moves through trees and over rooftops now, the oceans full of creatures with no common names because nothing remotely like them has been seen since. Into that world, or at least into the edge of that world, where life was elaborating itself rapidly and in many directions at once, the tardigrade lineage took its particular path. It kept its legs short and unjointed. It kept its body small. It kept the cuticle, the flexible outer covering that its ancestors had carried. The lineage did not become an arthropod, though some of the animals it was related to eventually did. It did not become a velvet worm, though the connection between tardigrades and velvet worms shows clearly in both the fossil record and the molecular data. It stayed, in some fundamental sense, close to where it started, small, wet, patient, occupying the thin films of water that exist in the spaces between other things. The fossils that connect tardigrades to that deeper Cambrian history are not tardigrades themselves, but animals called lobopodians, a group of soft-bodied creatures with unjointed legs, not unlike the tardigrade's stubby 8, whose fossils have been found in Cambrian deposits in China, Canada, and elsewhere. Lobopodians occupied the period before arthropod legs became jointed, before the exoskeleton hardened into the segmented armor that beetles and crabs and centipedes now carry. Their legs were more like lobes than like limbs. The tardigrade's legs, stubby and tipped with claws or adhesive pads depending on species, carry a structural echo of those ancient lobe feet across 500 million years of descent, a connection that is visible not in any individual fossil, but in the overall shape of the family tree, that molecular and morphological analyses have built, carefully and in pieces, over the past several decades. Tardigrades occupy a position within or near the group called Panarthropoda, the broader clade that includes arthropods and velvet worms, and the extinct lobopodians together, though exactly where within that group the tardigrade lineage sits has been the subject of ongoing discussion among researchers who study animal evolution. Some analyses place them closer to arthropods, others place them closer to velvet worms, the soft velvet-skinned animals with short paired legs that live in humid tropical soils and move with a slow, undulating walk that calls something to mind. The same patience, the same preference for wet and hidden places, the same indifference to being hurried. The debate is not resolved. The evidence is genuine and points in more than one direction, and researchers in different laboratories with different data sets have reached different conclusions, all of them careful, none of them yet conclusive. What is settled is that the two main classes within the phylum, Eutardigrada and Heterotardigrada, diverged from one another deep in the Paleozoic era, but a separation supported both by the physical differences visible in their bodies and by molecular phylogenetic analyses that push their last common ancestor back toward the early history of the group. The difference between the two classes is readable in the structure of their heads, particularly in the presence or absence of sensory structures called cephalic appendages, elaborate antennae-like projections in Heterotardigrada that are absent in the more simply built Eutardigrada. That difference, maintained across hundreds of millions of years of separate evolution, is one of the more durable illustrations of how conservative the tardigrade body plan has been, not frozen, exactly, since two distinct classes have clearly developed along different lines, but slow and incremental, and holding most of its fundamental structure intact across a span of time that encompasses multiple mass extinctions and the complete rearrangement of Earth's continental geography. The 5 major mass extinction events in Earth's history each eliminated a substantial proportion of the species alive at the time. The end Ordovician event, the late Devonian event, the end Permian event, the largest of all, which eliminated something in the range of 90 to 96% of all marine species, the end Triassic event, and the end Cretaceous event that ended the non-avian dinosaurs. Each of these left permanent marks on the fossil record, gaps where entire lineages simply stop. The tardigrade lineage does not stop. Its fossil record is sparse, because animals this small and soft-bodied do not preserve easily or often, but the molecular clock data and the Cretaceous amber specimen and the broader phylogenetic picture all point toward a lineage that was present before the first of those events, and that in some population somewhere, on some damp surface in some corner of the planet, that retained enough moisture continued. The ton state, that compact, desiccated configuration the animal enters when its water disappears, may have something to do with this. Small, dry, sealed, metabolically near silent, capable of wading through conditions that would end almost everything around it. The animal in its dormant form is not easily erased. There is a particular piece of Antarctic moss that sits in a research archive in Japan, collected in 1983, frozen solid from the moment of collection, and kept that way for 30 years in a laboratory freezer. In 2021, researchers thawed it and added water. The moss was gray-green and sparse when it had been collected from one of the cold coastal areas of Antarctica, where the growing season is short and the plant communities are low and small against the wind and rock. It had been kept at minus 20 degrees Celsius. Nothing about those 30 years of freezing would have struck anyone watching as temporary. And yet, when the sample was thawed and when water was introduced, tardigrades in the sample began, slowly, to extend their legs. The revival was not immediate. It took time, the kind of time that cannot be hurried, a gradual rewarming and gradual rehydration, and then incrementally the resumption of movement. 2 individual animals were confirmed to have revived successfully. One of them went on to reproduce, laying eggs that developed and hatched normally in the laboratory conditions. The offspring of an animal that had been frozen since 1983, since the year of their collection, on a piece of Antarctic moss on a cold coastal shore, completed their development without complication, as though the gap between their parents' last moment of active life and this one was a pause rather than an ending. The 30-year figure prompted researchers to consider whether the limit of viable preservation might be longer still under optimal conditions. The Antarctic environment, consistently cold and dark for long stretches of the year, offers conditions that could in principle extend the period of preservation beyond what any laboratory freezer has maintained. Specimens in permanently frozen soil, insulated from temperature fluctuation by depth, conceivably preserved by conditions that the natural world produced, rather than any deliberate archival intent. The question of how long such preservation could hold is genuinely open. No number has been established as a ceiling. The 2021 revival from the 1983 sample extended what had previously been documented, and the researchers who reported it noted that the new figure simply moved the known limit, not found the actual one. Sitting with that image for a moment, the frozen moss, the 30 years, the slow extension of 8 small legs into water after all that stillness, it is worth letting the time settle into something proportionate. 30 years is a long time in a human life. It covers the majority of a career, the growth of a child to adulthood, the slow accumulation of experience that constitutes something recognizable as a life. For the tardigrade in the frozen moss, it passed as nothing, or as something so close to nothing that the distinction may not be meaningful. The animal's body, in its dormant configuration, made no particular distinction between a month and a decade. It was waiting the way a stone waits, not actively, not with any awareness of duration, simply present and suspended, and available for the return of the conditions it needed. That capacity, the ability to wait across time without degrading, is not separate from the tardigrade's evolutionary history, but continuous with it. An animal that first appeared in the Cambrian, that survived the end Permian extinction, that was crawling across Cretaceous moss beds, while the amber that would preserve its relative was still liquid resin dripping from a warm tree. That animal's relationship with time is different from the one most creatures have, not in any conscious or experienced sense. simply in the sense that its biology has been refined across hundreds of millions of years of selective pressure to treat the passage of time under difficult conditions as something to be endured without cost. The machinery for doing this is built into the cells, into the proteins, into the specific molecules that the animal produces when its water begins to recede. It was not designed, it accumulated. It persisted in the population because the animals that had more of it survived conditions that ended the animals that had less, generation after generation, across a span of time that the fossil record can gesture at, but not fully render. The phylum that contains all of this, all 1,300 and more described species, all three classes, including the one represented by a single specimen from a Japanese hot spring that has since been lost, has been accumulating new formal descriptions at a consistent rate across each decade of study. New tardigrade species are not rare discoveries. They emerge from careful examination of samples collected from places where samples have not been collected before, or from re-examination of older collections using more precise tools, or from molecular analysis of populations that looked identical under a light microscope, but turned out to carry enough genetic divergence to warrant formal separation. The number 1,300 is not a final count. It is the current count, and it continues to grow. Most of those species are found exactly where the animal has always been found, in the thin films of water surrounding terrestrial mosses and lichens, on every continent, including Antarctica, in ocean sediments, in soil, in the wet surfaces of forest floors, on rooftop moss and garden wall lichen, and the damp interiors of cave systems. The habitat is defined not by geography, but by moisture and scale. Wherever there is a thin enough film of water, and wherever that film is associated with plant cells, or algae, or small invertebrates that can serve as food, a tardigrade may be present. The world, from this perspective, is an enormous and well-distributed network of tardigrade habitat, most of it invisible to anyone walking through it, all of it inhabited with the quiet consistency of an animal that has had 500 million years to find its way into the available spaces. The relationship between the tardigrade's deep evolutionary age and its current distribution is not coincidental. An animal that first appeared in the Cambrian has had an immense amount of time to spread, to differentiate into distinct species adapted to distinct conditions, to move with water and wind and the slow drift of continents into every corner of the planet that could support it. The 1,300 described species represent the accumulated result of that long, quiet radiation, a diversification that happened not through dramatic evolutionary leaps, but through the slow accumulation of small differences, species by species, environment by environment, across epochs that most animals did not survive at all. What the fossil record of the tardigrade lacks in density, it compensates for somewhat in precision. The amber specimens, and there are others, somewhat less well preserved than the 2021 Cretaceous example, preserve enough structural detail to anchor the living diversity of the phylum to specific points in geological time. 99 million years ago, in the Cretaceous, the claw structure that all tardigrades still carry was already present in its recognizable form. Whatever evolutionary changes have occurred since that moment have occurred within the constraints of that basic architecture, adding and adjusting and diversifying without abandoning the body plan's fundamental shape. This is not true of most animal lineages, where the passage of 99 million years tends to produce changes substantial enough that recognizing ancestor and descendant as related requires careful analysis. The tardigrade's conservative morphology Its tendency to stay close to its original form is one of the most consistent things about it. There is something in this that sits quietly alongside everything else the animal does. The tune state, the radiation tolerance, the vacuum survival, the pressure resistance, all of these are exceptional capacities, documented carefully in laboratory settings and real orbital environments, each one the result of specific molecular mechanisms built over evolutionary time. But underneath all of them, before any of them became significant or noticed or studied, there was simply the basic fact of the body plan continuing. The 8 legs, the rounded head, the slow rolling walk across a wet surface. These features did not require a mass extinction to demonstrate their value. They simply persisted, quietly, because they worked, because an animal built this way was able to find food and reproduce and survive the ordinary fluctuations of moisture and temperature that make up the everyday life of a small creature in a thin film of water on a piece of moss. The tardigrade that walked into Cretaceous resin 99 million years ago was not doing anything unusual. It was doing what tardigrades do, moving slowly across a wet surface, probably feeding on plant cells or algae, probably unaware in any sense of the resin that was closing around it. The animal that was revived from frozen Antarctic moss in 2021 was similarly unremarkable in its own moment of collection in 1983. One specimen among many in a sample taken from a cold coastal moss bed, preserved alongside others that did not revive, distinguished from them only by some small detail of its physiological state at the moment of freezing that allowed it to hold its cellular structure intact across 30 years of cold. Nothing about either animal in its own moment announced itself as significant. The significance is retrospective, the product of time, and of the patient observation of people who thought to look. Johann August Ephraim Gozzi, the German pastor who first described tardigrades in 1773, named them Kleiner Wasserbär, little water bear, because the slow, lumbering, 8-legged movement of the animal he saw through his primitive microscope reminded him of a bear navigating uneven ground. He had drawn water from a clump of dried moss and placed a droplet of it under his lens, and in that droplet, the animal was moving with the same deliberate rolling pace it carries through every description that has followed. He did not know its fossil history. He did not know that the claws he was looking at had been present in the same configuration in the Cretaceous, or that the animal he was watching would later be described as a polyextremophile, or that specimens of its kind would one day travel to orbit on the exterior of an unmanned spacecraft. He saw a small animal moving slowly across a glass slide, and he gave it a name that stuck. The name Lazzaro Spallanzani later added, Il Tardigrado, the slow stepper, is the one the phylum eventually took, Tardigrada. It is a name that describes the animal's most obvious and least dramatic characteristic. It moves slowly, not powerfully, not with any particular urgency, not in a way that suggests any awareness of the accumulated significance that later generations of researchers would find in its biology. It moves slowly across wet surfaces, feeding on what it finds, curling inward when the water recedes, unfolding again when the water returns, laying its eggs in the shed cuticle after molting, continuing the cycle it has continued, in one form or another, for longer than any of the continents have occupied their current positions on the surface of the Earth. A tardigrade recovered from a moss sample today, placed under a modern electron microscope, and imaged in detail, will show the same claws that the 2021 researchers saw in the Cretaceous amber. It will show the same circular mouth, the same band of muscle around the pharynx, The same arrangement of sensory structures on the head that vary predictably between the two main classes but remain consistent within them. The electron microscope makes visible what Gose's brass-tubed instrument could not resolve: the surface texture of the cuticle, the precise geometry of the claw tips, the fine structure of the leg attachment points. But what it reveals is not different in its fundamental organization from what has been present in the lineage for hundreds of millions of years. The technology for seeing the animal has changed enormously. The animal has not changed very much. That particular quality, the staying close to a form that works, the refusal to elaborate beyond what is necessary, is one of the quiet themes that runs through the tardigrade's long history. Many lineages that share the tardigrade's Cambrian origin are extinct. The lobopodians that it may have been related to, the Cambrian soft-bodied animals, whose fossil impressions are preserved in the fine-grained sediments of places like Chengjiang in China and the Burgess Shale in Canada, represent an extraordinary diversity of body plans, almost none of which persists into the present. What persists from that period of elaboration is selected and partial, the lineages that found configurations capable of surviving the long contingency of geological time, its extinctions and glaciations and volcanic episodes, and the slow shifting of the conditions that life requires. The tardigrade's configuration, it turns out, is one of those. It is not that the tardigrade is indestructible. Laboratory experiments have established where it fails, the conditions it cannot tolerate, the temperatures and doses and duration of exposure that exceed what even the tun state can accommodate. It is a real animal with real limits, not a metaphor for invulnerability. What it has is a particular set of capacities built through evolutionary time that extend those limits further than most animals have achieved, and a body plan conservative enough that those capacities have been able to accumulate and refine across an enormous span of time without being interrupted by the kind of fundamental restructuring that tends to discard what came before. The 2021 revival of the frozen Antarctic specimens was not, in the broader context, an exception to how tardigrades work. It was an illustration of it. The animal's biology includes the capacity to wait across hours, across years, apparently across decades, and to return from that waiting without measurable cost. The 30-year frozen specimen and the 99-million-year amber specimen are different kinds of evidence for the same underlying fact. The tardigrade persists, not through drama or urgency or any visible effort, simply by being built at the level of its cells and proteins and molecular machinery for the conditions it encounters. The Antarctic moss, where the 1983 specimens were collected, is still there. Presumably, the coastal moss beds of Antarctica continue to grow in their slow, low, sparse way through the brief polar summers, accumulating in the thin soil and on the exposed rock surfaces where the ice retreats far enough. In those moss beds, in the thin films of water that form during the warmer weeks and freeze again as the season ends, tardigrades are moving. Not the specific animals recovered in 2021. Those are gone now, their life cycles complete, but other tardigrades, their descendants perhaps, or other populations of the same species that have been living on that coast since long before anyone came to collect samples. They move at the same slow rolling pace that Goethe described, that Spallanzani named, that the Cretaceous animal moved across the wet surface it inhabited before the resin reached it. The question of how long a tardigrade might remain viable under preservation conditions that no laboratory has yet maintained, whether the 30-year figure represents a practical limit or simply the current edge of what has been tested, remains open in the scientific literature. Researchers who reported the 2021 revival noted the question explicitly. The methodology for testing it further would require samples maintained under controlled conditions for longer than the 30 years that separated collection from rehydration, and the logistics of that kind of longitudinal preservation study are not simple. The answer, when it comes, will come slowly, the way most answers about organisms that specialize in slowness seem to come incrementally through the accumulation of careful observations spaced across years. In a natural history collection somewhere, in the kind of long, quiet room where specimen drawers are arranged in order, and the light is controlled and the temperature is stable, there are slides and samples bearing the names of more than 1,300 tardigrade species, each one a label for a distinct version of the same body plan, each one representing a population of animals that has been or is living its small life in some film of water somewhere on the planet. The labels are precise and formal. The animals that gave rise to them were not. They were simply present in the moss or the lichen or the ocean sediment, moving slowly through their small world, feeding and reproducing and entering their resting state when the conditions required it, resuming when the conditions allowed. The tardigrade that is moving right now through a film of water on a piece of moss, on a garden wall, on a rooftop, on a forest floor, on the coastal moss beds of Antarctica during the brief summer, carries in its cells a history that reaches back to before the first fish developed jaws, before the first land plants, before the Cambrian seas closed over the Lobopodian ancestors, whose body plan it still faintly echoes. It does not carry this history consciously. It carries it the way any living thing carries its evolutionary past, in the structure of its molecules, in the arrangement of its genes, in the specific proteins its cells produce when the water begins to recede, in the exact geometry of the claws at the ends of its 8 stubby legs. It moves across the surface of the moss with the same slow, unhurried roll it has always moved with, pausing to feed, continuing, unaware of the amber specimen in the collection in the north, unaware of the frozen sample thawing in the Japanese laboratory, unaware of the long record of its own persistence that researchers have been assembling piece by piece from fossils and molecular data and laboratory exposure experiments and orbital missions.